Good morning, afternoon, evening, whatsoever may be the time there. So, composition and functions of the biological membrane. This is the first competency we are going to see. The biological membranes are present around all cells. They delineate all organelles in eukaryotes, and they are present. A simple design they have got, which is only two molecule thick. And this simple design is present in plasma membrane, intracellular membrane. Not only that, all species, all organisms, they have got this simple design, two molecular thick, and that is also referred to as the lipid bilayer. So the biological membranes are sheet-like structures, only two molecule thick, called lipid bilayer as we discussed just now because they comprise only of two layers and two lipid layers are there. And lipid also actually they are phospholipids. So phospholipid bilayer is the more appropriate term. Amphipathic in nature. Amphipathic ka matlab they have got a polar portion and they have got a non-polar portion within the same molecule. As you can see in this figure, this is the polar hydrophilic portion called the head group, and they have got a long hydrophobic or the non-polar tail. Head polar, tail non-polar, and an amphipathic molecule orients itself in such a manner that the polar portion faces water, extracellular or intracellular, and the non-polar portion is buried into the interior. That is understandable, entirely logical, because the polar hydrophilic water-loving portion will face the water, will interact with the water. Hydrophobic non-polar portion, it hydrophobia, it will try to go as much away from the water as possible. So therefore, that will be in the interior. So the bilayer structures, they are thin, fragile, like a bubble, but they form effective diffusion barrier called the hydrophobic barrier. Now, why do we say hydrophobic? You can see in this slide that hydrophobic portion is much larger compared to the small polar or hydrophilic portion. So they are mainly hydrophobic in nature, all these. As we said, around cell and organelle, and they restrict entry and exit of molecules. We will have separate lectures on that. And the two layer of lipids, they are non-covalent aggregates. They do not form any covalent bonds. And with embedded proteins, as we said, you can see here, proteins are embedded into it. This is the lipid bilayer structure. But note one point, even in this three-dimensional structure, it appears as if these portions are exposed to water, but that cannot be permitted. So they close onto themselves. I'll show in the next slide. Here you can appreciate in three dimension, protein, lipid core, and some amount of carbohydrate in form of oligosaccharide chain. Free carbohydrates are not present. So they close onto themselves. This is the way they close onto themselves to form, this is the, again, as we said, hydrophobic portion and hydrophilic portion. Perform various roles I have summarized in this slide. We'll be coming to details of various components in a short while. Various components are, as we said, lipid, which form core, protein for specialized function, and some oligosaccharides. Lipids are, mostly they are phospholipids. Now, phospholipids, the amphipathic molecules, as we said, they may be glycerol based or they may be sphingosine based. I will try to explain glycerol based. Glycerol is a 3-carbon compound. At the first and the second position, fatty acids are esterified. At the third position, it is a phosphate group and a polar base is there. So you can see this is the polar portion and this is the pole. This one is the non-polar portion, represented like this, polar and the non-polar. So amphipathic glycerol based. Sphingosine based का क्या मतलब है? Instead of glycerol, there is another amino alcohol. There is another compound which is an amino alcohol, and that compound is called sphingosine. It is an 18-carbon amino alcohol. So sphingosine 18-carbon amino alcohol, fatty acid esterified here, and then a polar phosphate and base. So sphingosine-based phospholipids are called sphingomyelins. What are glycolipids? This is the first, this is the second, this is the third type of lipid. In glycolipid, instead of sphingosine backbone will be there. Instead of this, an oligosaccharide will be there. So this is more appropriately called glycosphingolipid. Third, cholesterol. Cholesterol, it's a large molecule having this ring structure you will learn later on, and it has got an OH. जो भी है, it can be called amphipathic because hydroxyl group, it will face the exterior, just like in case of phospholipids. Fluidity of the membrane, we'll be talking later, is greatly influenced by cholesterol. So we have seen membrane lipids. What about proteins? Two types of proteins are there. Extrinsic protein called peripheral protein, integral proteins. I can show in this diagram. Extrinsic proteins are loosely attached to the surface of the membrane, whereas intrinsic proteins are actually immersed. They are more deeply embedded. A special type of intrinsic protein is this transmembrane protein, which traverses the whole length of the bilayer. And their functions, they perform enzymatic activity, recall brush border enzymes, which carry out the final stages of carbohydrate digestion. They are embedded in the brush border, lactase, maltase, sucrase. They are all integral membrane proteins. Carrier activities we'll be discussing in detail. They either act as channel or as transporter, which vary across various biomolecules. As we said, because of predominantly hydrophobic character, it does not permit flux of polar molecule across itself. So proteins ferry them across, transport them across the membranous barrier. Receptors for hormones and neurotransmitters. This again you will learn later on. Certain hormones, they bind to cell surface, and then certain intracellular events are influenced with the help of cyclic AMP, etc., called second messenger. So initially the hormone neurotransmitter, they would interact with the proteins. Then, of course, both function structural proteins are there. And finally, their interaction with extracellular matrix on the outside and cytoskeleton in the inside. So till now, we have done mosaic of globular proteins. Protein form this much percentage. Rest is mostly phospholipid bilayer. Various types of lipids are there, but exceptions are there. Proteins, they perform specialized role, all these. So wherever the metabolic activity is more, for example, in inner mitochondrial membrane, there protein may account for up to 75%. And myelin sheath, uske andar up to 80% can be lipids. Now we come to the third one, carbohydrates. This is just a summary of the functions performed by first two components. No free carbohydrates are present. They are attached to some protein or lipid, okay, on the external surface. And these are usually short chains of oligosaccharides, as you can see here. So glycocalyx is the name, hydrophilic carbohydrate layer. It forms a, if their functions now, they form a protective layer, protects the cell from digestion, involved in cell-cell recognition, adhesion, and receptor action. And finally, they impart asymmetry to the membrane. This again we'll be talking later. Fluidity, asymmetry, we'll be talking later on. So you can see transmembrane protein, just as a digression, not a digression, हमने जैसे बात की कि चैनल्स आर देयर इन द ट्रांसमेम्ब्रेन प्रोटीन जिससे कुछ आयंस वगैरह अंदर बाहर जा सकते हैं. So finally, fluid mosaic model, which is still widely accepted with some modification by Jonathan Swift and Nicholson. We have discussed bilayer organization with oriented lipids. We'll be talking, what do we mean by oriented? Embedded proteins. And hydrophobic it is because of long hydrophobic tails. Proteins may be intrinsic, extrinsic, and they perform a variety of role. Extrinsic protein may do read about spectrin. You'll be reading later on, but chalo, try. And then they are permeability barriers. We have discussed क्या होता है permeability barrier का मतलब do not permit everything to pass through. These three we'll be talking later just to give you an idea because I have repeatedly talked about fluid nature of the membrane. Membrane के अंदर lateral movement is possible. This lipid can go here, this can go here, here, here. Just like in a busy mall or metro station, people are traveling in all directions, but not all direction. Movement from one leaflet to another, that is not possible. Its role in deformability, mitosis, we'll be talking about later on. So finally, boundaries across the cell form compartments, act as diffusion barrier and sites of regulated transport involved in all these functions we have mentioned, associated enzyme we saw, fluid mosaic model finally views membrane as a two-dimensional structure of globular proteins and oriented lipids. So carbohydrates, they have got in form of, they are attached to lipids, glycolipids or glycoproteins. So this is all about it. We'll be talking more about those three things later on: asymmetry, fluid, and fragile nature with some clinical correlates. They will be mainly for the postgraduates and for the residents, but inquisite UGs, there is no bar to it. You can always see. Thank you.